never performed a single experiment. So even though he came up with the concept, the neuron doctrine, unlike Cahal, he had no hard experimental evidence to back it up. And what Cahal actually obtained were these beautiful line diagrams which he constructed from fixed tissue, from brain tissue. So here's the hippocampus. This is a slab cortex. You can see some of these cortical neurons, and here's a piece of retina all the way back to 1892. And basically the outline is entirely accurate, particularly for the hippocampus, and well, for all three, but particularly for the hippocampus, where you see the input fibers from the entorhinal cortex coming in, and you see these very fine branches of axons and dendrites. So his pictures, his diagrams have more or less endured the test of time over the years. So if you look at a single neuron, there are two basic points to consider. First of all neurons receive thousands of synaptic signals, inputs, on their very fine dendrites. So that's the input end. And these signals, called synaptic potentials, are exceedingly small in mammalian cortex. So from a single presynaptic terminal, when it releases transmitter in a mammalian cortical neuron, the synaptic potential is usually smaller than 0.1 millivolts. So in order to trigger an all or none action potential in the initial axonal segment, you need hundreds, sometimes thousands of simultaneously active synaptic inputs so that the synaptic potentials will actually summate to a value that's critical for initiating the action potential. Now, once the action potential is initiated, it can conduct all the way down to the presynaptic terminals where the action potentials will elicit the release of chemical transmitters. And the chemical transmitter will bind to the next neuron on specific receptors, and that's how you get a relay of electrical signals from one neuron to another. Just as a matter of detail, action potentials are more than 100 millivolts in size, baseline to peak, whereas individual synaptic potentials can vary tremendously, anywhere from less than 0.1 millivolts up to 10 or 20 or 30 millivolts. So you've got an input end and an output end for each neuron. A neuron that sends a signal is often called a presynaptic neuron. A neuron that receives a signal is called a postsynaptic neuron. And I'll be using these terms later on. Now, if you take a glass microelectrode and you penetrate a single neuron, what you find is that once the tip of the electrode is inside the cell, you will read a highly negative membrane potential, about -65, sometimes as high as -80 millivolts. And this membrane potential is known as the resting potential of the cell. And the reason the resting potential of a cell is negative is because the membrane at rest is highly permeable, particularly to one ion, and that's potassium. So it's the high permeability at rest to potassium ions that causes the membrane potential to be highly negative. There's a very low permeability to sodium. And also make note that the concentrations of sodium and potassium across the membrane are vastly different. So you've got high sodium on the outside, low sodium on the inside, and high potassium on the inside and low sodium on the outside. And in order for signaling to occur, ions must flow across the membrane. And in electrophysiology, we talk about ionic current, which is not the same as electron flow as in physics. In electrophysiology, all current flow occurs as a result of the flow of ions. And when positively charged ions flow out of the cell, we get an outward current, and vice versa. And you need ion channels, as I said before, which serve as specialized pathways to convey ions through the membrane. Without these ion channels, you would not have electrical signaling and you would not have brain function. So all of our brain functions depend on the existence of various types of ion channels in the membranes of nerve cells. So if this is the resting memory potential, and the memory potential swings towards a more positive value, say minus 70 to plus 10 millivolts, we call that a membrane depolarization. If the memory potential moves towards a more negative value, we call that a hyperpolarization. For example, minus 70 to minus 100 millivolts. If the memory potential, wherever it may be, moves back towards the resting memory potential, we call that simply a repolarization. Now there are three major types of voltage activated channels that are critical for electrical signaling. By electrical signaling, I mean the production of action potential. These three are shown here in a very simplified categorical diagram. They are voltage activated sodium, calcium, and potassium channels. Notice the opposite direction of natural fluxes. This is entirely the result of a concentration gradient pushing the ions through the membrane. Keep in mind that for each one of these categories, sodium, calcium, and potassium, there are several subtypes of each species. And I won't cover those subtypes. You'll see more of that if and when you take pharmacology 371. So let's take a closer look at one type, the prototypical voltage-activated sodium channel. So this is a voltage-activated sodium channel. It is selective only for sodium. Larger ions like potassium and calcium cannot pass through the channel because they're too large to squeeze through the narrow mouth. And the way the sodium channel works is as follows. At rest, the channel is effectively closed to all traffic because of an activation fence or activation gate that's shut. With excitation, if the memory potential becomes repolarized, this causes the activation gate to swing open, allowing sodium to flow into the cell. Now, if the membrane remains repolarized, what happens is you get a voltage-dependent inactivation, closure of the channel. And this results from a separate molecular entity, the inactivation gate, swinging shut. Notice the activation gate is still open, but now the inactivation gate swings shut to block the mouth of the channel. So this is voltage-dependent inactivation. And over time, as the memory potential repolarizes back towards rest, you get a recovery so that the inactivation gate swings open and very quickly the activation gate swings back closed. So the channel is back into its normal resting state, primed and ready for action for the next cycle of excitation. So this is a classic representation of voltage-dependent activation followed by voltage-dependent inactivation. It's not unique to sodium channels. It also occurs for potassium channels and also for some types of calcium channels. So let's talk a little bit about action potentials, because action potentials enable long-distance signaling. So the fact that you can sense pain in your fingertip is the result of action potentials traveling from your sensory nerve fibers in your fingertip all the way up to your somatosensory cortex. That's a pretty long distance. So you need a signal that's large and that will conduct without falling in amplitude, which fits the action potential perfectly. And Edgar Adrian was the first to record action potentials from single nerve cells way back in the 1920s and 30s. And as a result, he was able to formulate some of the basic principles of neural coding. And these are some of his actual recordings of action potentials. Notice the scale bars. Back then, they didn't have computers, so a lot of these axes were drawn by hand. And the shape of the action potential varied simply because of the way the recording electrodes were hooked up to the circuitry. Okay? So if you inject current into a neuron with one microelectrode and record with a second microelectrode, something which is very rare in mammalian neurons because they're way too small to squeeze two electrodes in. If you can do this, you can inject a square current. What you find is that most neurons, if the current is large enough, will elicit repeated firing of action potential. So these vertical strokes are, each one is one action potential. So you've got time versus memory potential measured in millivolts. And the more current you inject, notice the higher pulse of current, the more action potential you will elicit. So we say that the frequency of action potentials increases with the intensity of stimulation. You notice that the amplitude of each single action potential is fixed. So our brains cannot use action potential amplitude in order to distinguish between different stimuli. It needs to use a frequency code to differentiate weak from strong stimuli. And this principle applies all across sensory systems, visual system, auditory system, olfactory system, you name it. So what do we know about action potentials? Well, a lot of what we know first came about through the work of Alan Hodgkin and Andrew Huxley. And they pioneered the analysis of single action potentials in terms of which ions, figuring out which ions contributed to each phase of an action potential. And for their work, they were awarded the 1963 Nobel Prize. It's a typo in your notes. I think in your notes it says 1952. Their work certainly was done in the 1950s, but they won the prize in 1962. And they are basically to neuroscience and neurophysiology what Albert Einstein was to physics and his theory of relativity. The findings on the ionic basis of the action potential has held true all across the animal kingdom, including humans. So it's really a fundamental insight at the time, and it's contributed a lot to what we know about the physiology of neurons. So here's basically, in one slide, what Hodgkin and Huxley found. Here's a plastic action potential measured from a giant axon. You can see that a single action potential is quite short in duration, about 1 to 2 milliseconds, very brief, and quite large compared to synaptic potential. And we know that the upstroke, or the rising phase, is the result of sodium flowing into the cell through voltage-activated sodium channels. Okay? Once the action potential reaches its peak, it repolarizes, and the repolarization, or falling phase, is the result of potassium flowing out of the cell through voltage-activated potassium channels. So you get a sequential opening of voltage-dependent sodium channels, followed by a later opening of voltage-dependent potassium channels. You also get sodium channel inactivation up here, contributing to the repolarization. And the undershoot is the result of a hyperpolarization. Well, the undershoot is a hyperpolarization that results from slow recovery of potassium channels, slow removal of potassium channel activation, or if you like, slow potassium channel inactivation. And eventually the memory potential relaxes back to the resting potential. So that's the ionic basis of the action potential. And the principle here holds true all across the animal kingdom. And I think even some plant cells show this action potential. I'm not sure. Why would they? Who knows? So not surprisingly, if you pharmacologically block sodium channels, you basically interfere with action potential initiation. And in this way, you can kill pain. Anesthetics, commonly used anesthetics, such as Novocaine and lidocaine, local anesthetics, their mechanisms of action all converge upon one single unitary mechanism, and that's sodium channel antagonism. It's a voltage-dependent sodium channel block. General anesthetics used to put patients to sleep for surgery. Some of them potentiate inhibitory transmission by enhancing inhibitory receptors, such as ABA receptors. And the commonly used one is propofol, which looks like a milky white liquid. And some patients, not all, but some patients report post-surgical amnesia, temporarily, of course, after waking up from propofol in the recovery room. And as a result, the surgeons joke among themselves that propofol is actually the milk of amnesia. I mean, a long time ago, they had an acid called milk of magnesium. It might still actually be available on the shelf, but I think they took it off the shelf for years now. It's called milk of magnesium. So that was used as an antacid to neutralize stomach acid, basically to treat heartburn. So surgeons call propofol the milk of amnesia because in some patients there's a post-surgical report of memory loss. Now, axons are often myelinated, and myelin is a fatty substance that wraps around sensory and motor axons. And basically, the myelin acts as an electrical insulation, much like the insulation on an electrical cord. So what it does is it reduces the amount of current ions that leak out of the axon membrane as the action potential is shooting down the axon. Okay. Now, not the entire axon is wrapped in this sheath. There are regular interruptions, tiny nodes of Ranvier, called nodes of Ranvier, where the membrane is basically naked, uncovered by myelin, but where there's also a very high concentration of voltage-dependent sodium channels. So it turns out that these tiny nodes of Ranvier, which occur at regular intervals, act as sort of like boosting stations to boost an action potential as it spreads along the axon. So as the action potential spreads along the axon, in between the nodes, there's a little bit of reduction in amplitude as some tiny amount of current does leak out. But by the time the action potential reaches the next node, there's enough voltage to really activate sharply the sodium channels, and the sodium channels will actually replenish the amplitude, reboost the amplitude of the action potential. So as a result, you get a faster conduction of action potentials along a myelinated axon than you would without the myelination. But this is called saltatoric conduction, where the action potential appears to jump from node to node. And that's the essence of saltatoric conduction. So with the myelin sheath, you get a faster conduction velocity, so it takes less time for an action potential, for example, to reach your somatosensory cortex from your thumb or from your toe than it would without the myelin. Now, in cases where the myelin sheath is actually ripped away because of neurological disease, you lose that insulation and you get a slowing of conduction and a slowing of limb movement in the case of multiple sclerosis. So the essence of multiple sclerosis is that the patient's immune system produces antibodies that attack their own myelin for reasons not fully understood. And over time, the myelin sheath gets ripped away, conduction velocity slows, and you get a slowing of movement in the limbs. And eventually the person probably ends up in a wheelchair in the most advanced stages of MS. And here are some of the symptoms and possible mechanisms of MS. Again, here are the antibodies. They attack the myelin. Eventually, over time, the axons get stripped away to an inflammatory process. When this happens to the optic nerve, you get a gradual loss of vision. And you also get loss of control of your urinary bladder and other motor disorders, such as ataxia, which is a lack of muscle coordination. But usually the first signs of muscle weakness followed by some very often optic inflammation of the optic neuritis. Are there any questions so far? Yes, sir.
Potential before an action potential is minus 60 millivolts.
Yeah.
Is that the natural potential that is set all the time?
Well, the resting potential, so the question was what's the range of resting potential values for nerve cells? It can range anywhere from minus 60 to as low as minus 80 millivolts. That's a resting potential. So when that membrane potential reaches a critical threshold value, say minus 10 or minus 20 millivolts, you get an explosive all or none action potential. Okay. So enough about action potentials. I now want to move on to chemical synaptic transmission. Action potentials will trigger the release of chemical transmitter at nerve terminals. And these chemical transmitters will be released into the synaptic cleft, the tiny space in between the terminal and the receiving neuron, the postsynaptic endoid. So the telltale sign of a chemical synapse, of course, is the presence of these vesicles in the nerve terminals that contain the transmitter substance. Another telltale sign is this tiny little synaptic space called the synaptic cleft. With electrical synapses, the two neurons, presynaptic and postsynaptic, are actually in physical contact with each other. There is no synaptic cleft. So with electrical synapses, you get a very, very rapid spread of transmission from one cell to the next. And these electrical synapses are specialized for synchronous activity among large populations of cells. For example, your heart muscle cells, cardiac cells, are connected to each other by electrical synapses through junctions, gap junctions. And that's how your heart muscles can synchronously contract together. With chemical synapses, because of this tiny synaptic cleft, that introduces a tiny amount of time required for the signal to jump the gap from one neuron to the other. So you've got to mind the gap in chemical synapse. And the first person to report chemical transmission was this man, Henri, a German pharmacologist. And he did a pretty clever experiment shown right here in this sample trace. So this is a tracing of muscle force. Each vertical line here on the compressed time scale represents a single contraction. The higher the line, the more forceful the contraction. So what he did was he electrically stimulated the vagus nerve from one frog heart, collected the secretion, and then transferred or perfused that secretion over a second heart, shown here on the arrowhead. So when he did that, the contractions were reduced in force. Okay? He then activated the sympathetic nerves to that heart, frog heart 1, collected the secretion from that nerve. He didn't know what the secretions were at the time. Collected that secretion and perfused the second heart with the sympathetic secretion. And what he saw was the opposite. With the sympathetic nerve secretion, the second heart increased its force of contraction. So this was the first proof that the nerves were releasing some chemical transmitter that could convey excitation or inhibition to a bunch of excitable cells, heart cells in this case. We now know, of course, that the vagus nerve and the sympathetic nerve secrete. Anybody? Lennon Watson. Good show. acetylcholine, and norepinephrine. Another major figure in chemical transmission is Bernard Katz. In the 1950s and 60s, Katz and his colleagues performed some amazingly elegant experiments that I don't have time to show you, in which they showed through some very technically sophisticated experiments that calcium entry into the nerve terminal was critical for the release of chemical transmitters. They also went on to basically break down the process of chemical transmission in a quantitative manner. So thanks to them, we're now able to analyze changes in synaptic strength in a quantitative manner without relying on qualitative methodology. We can put numbers and statistics on such parameters as the probability of release under certain conditions, thanks to Bernard Katz. Katz and Lowy, for their work, won respectively Nobel Prize in physiology and medicine. So some of you might be wondering, do you need to know these individuals? My answer is yes. Okay. Science is done by people, and you got to put a face and name to the work. It's not anonymous work. So this is one of my heroes, Steve Kupfer. I never met him. I read about his work. I used his textbook, From Neuron to Brain. The first and second editions were originally from him. It's now in its 6th edition or something, and it's the current textbook for Zoology 342. Kupfer, he was a Leonardo da Vinci of neuroscience. I call him the Leonardo because he didn't stay put in one field. He was restless, jumped all over the place, and as a result, he came up with these original pioneering discoveries. He was the first to show that neuropeptides could excite neurons. We now know that there are tons of neuropeptides. First to show that glial cells could communicate with neurons. Figured out the physiology of neuromuscular synapses. The stretch receptor, which some of you might have encountered in your physiology courses, he was the first to discover that using crayfish. He originated the concept of receptive fields, particularly in the retina. And that concept extends now to every sensory receptor we know of, auditory receptors, touch receptors. He was also the first one to create a department of neurobiology at Harvard. In the 60s, there was no such thing as neurobiology. I mean, the idea that in one department you could get many individuals with specialist degrees in neuropharmacology, neurochemistry, neurophysiology, neuroanatomy, all working under one roof in a department. That was alien in the 1960s. Nowadays, every major research university has a department of neuroscience, or in our case, even better, a center for neuroscience. So he was the first to create a department of neurobiology, which still exists at Harvard. And of course, the textbook from Neurons to Brain. This is Steve Kupfer. This is David Hubel. Hubel and Wiesel won Nobel Prizes for their work on the visual cortex in cats and monkeys. Hubel was born in Windsor, Ontario. Moved to the States, where he's been a professor at Harvard's Department of Neurobiology since the 60s. And he's one of the nicest people you can talk to, very low-key despite his enormous contributions to being a Nobel laureate. You generally find that Nobel laureates are very nice, contrary to popular belief. Because they realize once you win the Nobel Prize, you're under a microscope. People expect you to be right all the time. So the prize of anything humbles them quite a bit, which is good. So let's talk a little bit more about chemical synaptic transmission. Again, this is a chemical synapse, mind the gap. Presynaptic cell forming terminals onto the postsynaptic dendrites, the receiving end of a postsynaptic cell. Of course, every neuron can be both presynaptic and postsynaptic. It's just a relative term to convey which cell we're talking about here. Here's the sequence. I want you to know the sequence, inside and out. It's, I think, fundamental and logical. You get an action potential reaching the nerve terminal. This causes a membrane depolarization. that opens up voltage-activated calcium channels. The calcium rushes in through its concentration gradient, and it does something to cause vesicles to fuse with the membrane and release the transmitter molecules into the clot. And it takes less than, let's say, 100 microseconds for the transmitter to float across the tiny gap, where it binds to specific receptors, which then either open up or sometimes close channels. So as a result, you get ion flux through these opened transmitter activated channels in the postsynaptic cell. And these ion fluxes can then lead to either excitatory or inhibitory postsynaptic potential. So in the mammalian brain, including the human brain, the major excitatory transmitter is the amino acid glutamate. And when glutamate is released, it basically binds to two major classes of receptors: AMPA receptor and NMDA receptor. We're talking only fast synaptic transmission here, not slow transmission, which involves other classes of receptors, which I don't have time to talk about. Now, you notice the naming of these receptors. They're AMPA and NMDA receptors, but they both bind the natural transmitter glutamate. Don't ever think that they bind AMPA and NMDA in the brain. There's no such thing as AMPA and NMDA in the human brain or in any mammalian brain. Those are synthetic compounds that were used initially to identify the receptors way back in the 1970s and 80s. Now, when the cell's at rest, the AMPA receptor can conduct predominantly sodium ions to produce the EPSP. At rest, the NMDA receptor, even though it binds glutamate, it can't conduct because normally NMDA receptors are blocked by magnesium at rest. So the magnesium block turns out to be pretty important. In order for conduction to occur through NMDA receptors, you have to have a strong membrane depolarization to unblock the magnesium, basically to knock the magnesium out of the channel. And only then will you get flow of calcium and sodium through the NMDA receptor. So two conditions need to be satisfied for NMDA receptors to conduct. First, you need to have presynaptic activity to release the glutamate, because remember, you still have to have glutamate binding to the receptor. And simultaneously, you need to have postsynaptic membrane depolarization to unblock the NMDA receptor. So in other words, the NMDA receptor is very good as a coincidence detector. When it's active, it basically enables the cell to know that both presynaptic activation and postsynaptic activation are occurring simultaneously. And when this occurs, when NMDA receptors are activated, you get all sorts of interesting forms of synaptic modification, such as long-term potentiation, which has been linked in some cases to learning and memory. So at mammalian synapses, when you get glutamate release, you get an EPSP, excitatory postsynaptic potential. And if the EPSP is large enough, it will elicit an action potential. On the other hand, you can inhibit the postsynaptic cell if the transmitter happens to be GABA, which is another modified amino acid. And in the mammalian brain, chloride channels are opened when GABA binds to its receptors in the postsynaptic spiral. And when chloride flows into the cell through its concentration gradient and down its gradient, you get a hyperpolarization or an IPSP. And this is inhibitory because you're moving the membrane potential farther and farther away from threshold, which is further up, more positive. So anything that moves the membrane potential away from threshold will reduce excitation. Will suppress part. All right. So now we come upon ligand-gated, chemically activated channels. And there are many types. I just told you two types, AMPA and NMDA receptors, both of which bind glutamate. There are also kainate receptors and metabotropic glutamate receptors, both of which also bind glutamate on a slower time scale. There are also other channels which are chemically sensitive, calcium activated potassium channels, which let potassium flow out of the cell, but which are sensitive to calcium. They need to have calcium bound to it in order to activate the channel. There are also cyclic nucleotide activated channels that bind either cyclic GMP or cyclic AMP to open a channel that's permeable to more than one type of ion, sodium and potassium. Now, what are some of the psychiatric disorders associated with disturbances in specific transmitter systems? I'll show you only three here: depression, schizophrenia, and anxiety disorders. And basically, if you know the basic underlying cause, you can sort of figure out what the logical therapeutic strategy is. So in the case of depression, this has been associated with a sharp reduction in the levels of serotonin and sometimes noradrenaline, norepinephrine. And not surprisingly, antidepressants act to elevate serotonin or norepinephrine by inhibiting reuptake of the transmitter. So it allows the transmitter to float around in the synaptic cleft for a longer period of time. Schizophrenia, we don't quite know the cause of schizophrenia, but one common factor is excessively high levels of transmission through dopamine. So a lot of the drugs that have been used to treat schizophrenia, not surprisingly, antagonize specific types of dopamine receptors, particularly D2 type. And anxiety disorders such as PTSD are generally treated with drugs that enhance cortical inhibition by potentiating GABA receptor function. These are drugs like Valium, which is a benzodiazepine. Also by, in some cases, drugs that block reuptake of serotonin, such as Prozac. The serotonin generally has a relaxing effect on people. So now let's get into the software of the brain. I like to call this the software, the intracellular signaling. So far we've talked about how signals are transmitted from one neuron to another. That's called intercellular signaling. Now I want to talk to you a bit about intracellular signaling, the signaling that occurs inside a single neuron. So here you have on the cell membrane your first line of offense, receptor and a transmitter, and also the G proteins that are coupled to the receptor. And these components increase the levels of second messengers. And right now there are only 5 known second messengers in eukaryotic cells, including nerve cells, and they are all shown here. 2 separate nucleotides, 1 ion, calcium, a lipid, and inositol triphosphate, which happens to be a carbohydrate. It's not a lipid. These second messengers can engage third messengers, which are protein kinases, which when activated add phosphate groups to protein. Now what goes up must come down, so there has to be an antagonist, and these are protein phosphatases, which remove phosphate groups to switch off phosphoprotein. And as a result of these third messengers, you get multiple processes occurring in the cell on various time scales. You get very fast ion channel activation, inactivation, all the way down to persistent processes like synaptogenesis and learning and memory, which can persist for an entire lifetime. Now, I'm not implying that phosphorylation can last an entire lifetime. It can't. But the processes that are regulated by phosphorylation can last an entire lifetime in some cases. And these are separate processes that may rely on phosphorylation initially as a trigger, but that can be sustained through other mechanisms like transcription and protein synthesis. So here are some general properties of signaling, the software, the operating system. 5 properties. I want you to know them. I won't go through them. It's all there in detail for you. A lot of them are sort of common sense. And here's a long laundry list of the major kinases and signaling molecules in neuroscience. I want you to know these 4 kinases. They are the common vocabulary of neurobiologists nowadays. You'll learn them sooner or later. I think a lot better sooner. I added this morning. Don't panic. It's a website that lists a lot of kinases and their associated diseases. I won't test you on it. So for example, ERK, too much ERK activity has been linked to certain types of breast cancer and prostate cancer. Too much PKC activity has been linked to many types of cancer, too many to mention. CAM kinase 2, in particular, has been linked to cardiac arrhythmias. And PKA, in a paper published just last year, you're getting the most up-to-date information now, has now been shown to be reduced in expression in the frontal lobes of children with regressive autism. They didn't find it in other lobes, just the frontal lobe, post-mortem analysis. So in a nutshell, kinases, or rather tight regulation of kinase activity, is crucial for normal health. Too much kinase activity and you often end up with cancer, which makes sense because kinases regulate cell growth. You get too much kinase activity, you get a loss of control of cell growth, which is the definition of cancer. Now it might seem at first sight that, well, why not cure cancer by targeting the kinases? I wish it was that simple. There's a joke among cell biologists that they've cured cancer years ago. The problem is they've cured it in mice. There's something fundamentally more complex with human cells versus mouse cells, but we just haven't come to understand fully yet. There are phosphatases, the 2 major ones are listed here, and then there are these proteins that can be phosphorylated. Among them, NMDA and antireceptors. So let's talk a little bit about G protein. This is a G protein. It consists of 3 subunits, alpha, beta, and gamma. When it's activated, it will bind GTP, it will convert GDP into GTP. And in some cases, it's the beta gamma subunit that binds to the target protein, for example, some types of potassium channels, to open the channel. In other cases, it's the alpha subunit and not the beta gamma subunit that binds. For example, the enzyme that produces cyclin AMP, adenocyclin. It's activated by the alpha subunit from the G protein, binds to it. Now, of course, this signaling pathway involving the intermediate G protein takes a little bit more time than if you had no G protein. But it also introduces an opportunity for modification of plasticity. So the price you pay in time, I think you recover in terms of the opportunity for fine-tuned regulation of plasticity, because you're introducing an additional target for regulation, the G protein. The man who discovered the first sucker messenger, historically the first sucker messenger, sucker AMP, is Earl Sullivan. And since then, sucker AMP has been shown to be important not just in nerve cells, but in practically every eukaryotic cell, apart from red blood cells, particularly cells that secrete hormones, endocrine cells. So let's take a closer look at this PKA pathway because it's my favorite molecule. Everyone has a favorite molecule, right? So mine's PKA. Spent much of my career examining the role of PKA in learning and memory. So here's PKA. it exists as a 4-subunit enzyme. 2 of them are regulatory, 2 of them are catalytic, the green ones. It's the catalytic subunits that are actually phosphorylate proteins. And the C subunit can only do this, phosphorylate, when they're released from the R subunit, the regulatory subunit. And the R subunit will bind to the C subunit to hold them in check until cyclic AMP increases and binds to the R subunit. So cyclic AMP increases pKa activity by releasing the inhibitory regulatory subunit. And once released, the activated pKa can phosphorylate many proteins, one of which is CREB. CREB is a transcription factor found only in the nucleus, it's not in the cytoplasm. And together with the helper protein called CBP, CREB can assist in the transcription of particular genes, some of which are shown here in the lower right-hand side of this slide. And once transcribed, the transcript gets moved out to the cytoplasm where it can be translated into these proteins. And there are 2 disorders that result from mutations in CBP or underexpression, reduction of pKa. They are Rubinstein-Thibault syndrome, which is a form of mental retardation. So if you mutate, if CBP happens to be mutated, you often will get Rubinstein-Thibault syndrome. If pKa is reduced in activity in the frontal lobes of infants, you get regressive autism. And this is evidence from a paper just published last year. Postmortem analysis of a small group of brain tissue from human patients. So with regressive autism, the child initially develops normally, but after about 15 to 30 weeks, the child begins to show signs of social maldevelopment, signs of autism. Now, whether or not this is an exact cause, we don't know, to decrease pKa, but it is tempting because the group showed it only in the frontal lobes. They looked into the other 3 lobes and they found normal levels of pKa. So this is a region-specific decrease in pKa. And remember, the frontal lobes are critical for social behavior. And one of the hallmarks of autism, as you'll learn in a later lecture, is social maldevelopment, impairment of social interaction. So I'm a bit excited about this finding. One of very few neurological disorders to be sort of linked to pKa at this time. So how do we know what we know about the roles of genes and proteins in brain disease? We know because a large amount of research is performed on animal models, particularly mouse models of brain disorders. And transgenic mice are mice in which a transgene that encodes an endogenous protein is introduced into the genome. And to do this, you attach the coding sequence of the gene you're interested in to the promoter for a protein called CAM kinase 2 alpha, the alpha subunit. And it turns out that the alpha subunit of CAMK2 is expressed only in brain tissue. So if you attach the promoter of that subunit upstream of your gene of interest, and then inject fertilized eggs with that transgene and reimplant these fertilized eggs into the uterus of the female mouse, some of its offspring will carry the transgene will express the transgene only in brain tissue. Other offspring will be wild types without the transgene. And once you identify the transgenics, you can do lots of neat things with it. You can do brain slice electrophysiology to look at synaptic changes. You can probe levels of expression across multiple brain regions to see exactly which brain region has the highest level of expression of the transgene. And you can do behavioral analysis, which I'll talk about a little bit later on. So if you look at the pKa transgene for the regulatory subunit, Ted Abel, a longtime collaborator of mine, managed to get the gene for the regulatory subunit mutate it so that even though it binds sec-AMP, it fails to release the catalytic subunit. And when he expresses this in mouse brains, what he finds is that it reduces basal PKA activity in the hippocampus by about 40%. So you still have a fair amount of PKA there. So he was lucky. It wasn't a lethal expression because you still had a substantial amount of PKA there. But what's remarkable is that this was enough to impair long-term but not short-term memory in these animals. This cartoon simply shows what a mutant RF subunit will do. They bind sec-AMP, but they don't release the catalytic subunit. This is the expression pattern in an RGA mouse and in a wild-type mouse. This is the front of the brain. That's the cerebellum. That's the hippocampus. Notice how intensively it lights up with the mRNA probe for the transcript. And in humans, these are all the different possible types of subunits, R and C. Don't worry about this, I won't test you on it. There's 60 possible combinations of these 4 subunits. And remarkably, the concentration of PKA in mammalian cells can be as high as 2 micromoles. This is for a second mechanism. And one of the enduring principles from this work, as well as from earlier work, is that long-term memory, without short-term memory, requires the synthesis of new proteins and transcription. Intermediate-term memory, lasting only minutes, requires pre-existing mRNA and not new transcription. And again, the focus here is on CREP, so let's talk a little bit about CREP in the time that we have left today. CREP's a remarkable molecule, like kinases. Too much of it or too little of it can lead to drastic consequences. And CREP has been implicated in addiction, drug addiction. And I like to think of CREP as sort of like a cognitive crazy group. Once the bond is formed, it's really hard to break the bond. The bond I'm talking about here is the memory, long-term memory, or association between a substance of abuse that gives pleasure and the behaviors that you had to undergo to get more of that substance that gives pleasure. And once that association is formed, addiction ensues. You crave more of the drug, and you remember the behaviors that you need to go through to acquire more of that pleasure-inducing drug. Not surprisingly, you can't treat addiction with drugs, right? The person will become dependent on the therapy. Instead, drug addiction is commonly treated through therapy involving behavioral training, behavioral therapy. Not psychotherapy, which is different. Behavioral therapy. And the critical brain structure involved in addiction is the nucleus accumbens. And the transmitter that's most often associated with addiction is the transmitter dopamine. Dopamine is known as the pleasure transmitter. When we get pleasure out of something, our dopaminergic pathways fire like crazy. With drug addiction, for example, with cocaine and amphetamine, your brain pathways get used to seeing high levels of dopamine, abnormally high levels of dopamine. And because these pathways are accustomed to that, you're constantly seeking ways of attaining and hyperstimulation of dopaminergic pathways. Now, over time, the neuron adapts. It actually reduces the number of dopamine receptors. But the levels of dopamine are still pretty high. And the key to fixing drug addiction is to break that link, break that crazy glue, cognitive crazy glue link. Possibly through manipulation of CREP or some other strategy. The problem with manipulating CREP is that CREP is involved in so much more that you can't target it to treat just drug addiction. There are non-specific effects of messing with the molecule of CREP. But you can target CREP to look at processes such as learning and memory. And Sheena Joshua's lab showed years ago that if you knock out 2 isoforms of CREP, the alpha and delta isoforms in the brains of mice, and assess long-term memory for contextual theory, you can impair long-term memory. So with contextual theory, you place the mouse into a box with a metallic floor. The mouse receives a very brief foot shock. And a normal mouse remembers the association between that box and the foot shock. So after a time delay, if you put the same mouse, the shocked mouse, back into the same box and just leave it there for 5 minutes and measure the amount of time it spends completely frozen with fear, you can have an index of contextual fear memory. So let me show you a video here. Some of you have seen this video already in previous classes. So anyway, this video shows 4 mice. 2 of them are wild type, 2 of them are crab knockouts. They were placed back into the box 24 hours, which is long term for mice, 24 hours after the shock training session. So all 4 have been shocked, and this is a 24-hour test. Now the wild types with so-called normal memory should be completely frozen because they recognize the box as having been previously shocked in. The knockout mice should be moving around as if the box was brand new to them, which it isn't, of course. So let me play this. 2 of them are wild type, 2 of them are knockout. So the results are pretty dramatic. This is the best possible case. Turns out these 2 are knockouts. These 2 are the wild types. Notice how they're frozen in the corner, typical fear posture for a mouse. And these guys are moving around like it was a novel environment. So this is 24 hours after being shocked. Now the critics, the sharp people in the audience will say, well, how do you know the knockouts aren't just smarter because they're looking for a way out? And my answer to that is, unfortunately, I don't have the video, but if you do this test on 1 hour, with the short-term memory, all 4 are moving around. Sorry, all 4 of them are actually frozen zones. So because of that, we believe that long-term and not short-term memory is impaired. All right, that's the end of my lecture.